Without further ado, ladies and gentlemen, please help me welcome to the stage 2007 Nobel laureate, Dr. Mario Capecchi.
First of all, that's a terrific introduction. And, you know, the reason I'm here are for the students. I mean, I think you are the next generation. We've created a lot of problems for you, and you're going to have to solve them. So, you have to be tenacious. You have to feel, go with your gut and just do it. We've been too timid, and now the time has come for you. It's your turn. And I hope you do a much better job than we did. So today's story, I'll just put it together and we'll see how it works. And I think it's an unusual story. Now, you know, the reason to be in science is you're always surprised. Mother Nature is an amazing woman. I mean, she's really amazing. And she doesn't think necessarily logically, but what she thinks about is that things work. And when it works, then she just keeps repeating it and utilizing it in different contexts. And I think what we'll see here in this story is, again, the wonders of what Mother Nature has created. She's had a long time to do it, and she's really, really clever. And often we underestimate what she is capable of doing. And I think that's what makes science exciting. So let me get started. Let's see, we've got a bunch of contraptions here. For a long time, this is the Ocd genes. And for a long time, our laboratory has been studying. And they're an interesting set of genes because they're involved in making the body plans of all animals, all multicellular animals, including us. Right now, I'm showing you Ocd genes from mouse and humans. It's an identical cluster. It's 39 genes. Wherever you see a box, it's a gene. And wherever you don't see a box, there used to be a gene there, but then it wasn't needed anymore. And so you can still see the traces of the gene there, but it's no longer functional. And different people in the lab, we've inactivated all of these mice in mice. I mean, we've inactivated all these genes in mice. And we're studying exactly what each one does. And Joy Greer, a very talented student in my lab, graduate student, her job was Ocd. And she got it, you know, most of these genes are, you know, they make sure that our head is at this position, our arms are at this position. They essentially form an axis to tell you where everything in your body should be and where you should start making those parts. And then you make them, join them all up together, and you have a functional human being. Joy Greer's job was to do Ocd. She inactivated it in a mouse. We developed the technology to change any gene we want in the mouse and see what happens. She did that, and she got a real big surprise. The mouse was perfectly normal with respect to body shape. She didn't expect that. But what was changed was the behavior, what the mouse was doing. So what she did was simply take a camera, and it was an infrared camera because mice do all their activity most in the dark, at nighttime. So she takes 24 hours of movies and then starts jotting down, frame by frame, what is this mouse doing? And she studied everything possible, moving around in a cage, walking, eating, drinking, building nests, walking upside down in a cage. They can go out on top. They can do anything, okay? And she just jotted down every time they were doing something, put a little X, and that associated with how much time. And she looked at this particular gene, and the only thing it was doing different is how much time they spent grooming. And the grooming turns out to be very important, okay? It's a way of getting rid of pathogens. It turned out that it increased our lifetime over 20 years in the Middle Ages, when they changed essentially from living in wool, which you couldn't wash, to living in cotton, which they could wash. They started washing their clothes, and then they started washing themselves, and now they live 24 years. Well documented. But in this, these mice had too much grooming. They spent a lot of time grooming. And it so much so that it became pathological. You may know somebody that has OCD, obsessive compulsive disorder. They wash their hands maybe over and over again. They don't get signals to say, my hands are clean, and they just continue to wash until it's pathological and they have lesions in their hands. Okay? So it is pathological. And it's called trichotillomania. And it's very common. 3% of all populations in the United States, 7 different populations have been looked at, between just a little under 3% to over 3%. So in this audience, you can, there's 7. Okay? It's very common. And they remove their body hair, all their body hair. And here is, I told you all animals groom, coelacanths, little worms groom, Drosophila fruit flies groom, all mammals groom. And the syntax, the pattern of grooming is similar in all animals, in all mammals. Okay? You go to the shower, you lather your hands with soap, and you start working on your head, and you go down your body. And if there's anybody in this audience that has a different pattern, please see me afterwards. I'm going to show you a little movie about the grooming. You can see that the mouse just keeps grooming and grooming and grooming. And what you'll see is it keeps on grooming until it actually grooms part of its, when it's accessible, which is the tummy side, and starts removing its body hair. You'll see. It turns, and there's all the hair's gone. And then it continues to groom until it has lacerations, and at that point we put it to sleep. Okay? Now I'm going to describe the pattern of this behavior very carefully to you and in great detail. And what's fascinating is even in this minute detail, it's identical to humans. The behavior is corresponding. You'll see many different patterns, and you'll see that it is identical to humans. This shows you, first of all, it's a control mouse. Okay. That's a normal mouse. And this is a heat map, and what we do is simply put on top of one image after another of the grooming pattern of one mouse after another. And then where you get more and more overlap, then the color turns yellow and finally white. And so that is where the pattern's always similar, and then as it gets more rare, it'll become lower down to purple. Okay? So this gives you a heat map. It shows you that females groom more than males. Everybody knows that. And this actually quantitative. So here we're looking at females. Normal doesn't remove its body hair, and then that level for a mutant, and the gene is HOXB8. So we inactivated that gene, and the only thing that's different about that mouse is that behavior. Okay, and then we look at the null mouse here with respect to grooming time, and here how much code is lost. And males, this is a slightly different scale, so males do it less, and they also spend less time grooming. Okay? But nonetheless, they still go into the pathology. And here you directly compare males and females, and you can see that it's much more prevalent and aggressive in the females. Another feature of this mouse is that it has high levels of anxiety. I think all of you know about anxiety. You get anxious about something. If you are anxious, then you go certain patterns. And it turns out that these mice are also anxious. And people with trichotillomania are also anxious. What they tell me, I've interviewed over thousands of patients, and when they're anxious, then they go into their malbehavior. When they're not anxious, they can control that behavior. Okay. So, and with mice, we have multiple ways of assessing anxiety. Okay? We can put them on a high platform, and one bar, there's two of them crossed each other. One has walls up it, and the other is open. A normal mouse would go in both bars equally well, looks down, doesn't worry. An anxious mouse will avoid the open one that doesn't have walls there. Okay? And that's one level of anxiety. And here we see essentially normal, and then wild type. Here it's never going, it's avoiding the open arms. Normal, and then HOXB8 mutant mouse, never going to the open arms. So that's one way of measuring anxiety. There's another simpler way. You simply put a mouse on a field, a big field, and then a normal mouse will go all over and explore the whole area. An anxious mouse will stay out in the perimeter where the walls are and never go into the center. That's shown here. So this simply traces where the mouse has been. A normal mouse goes everywhere. Normal means wild type. A mutant mouse stays away from the center. Wild type again, everywhere. Mutant mouse stays away from the center. That's another way. Another simple thing, you can simply give two chambers. One has a light in it and the other one's dark. A mouse, because it does most of its activity at nighttime, prefers the one that doesn't have a light on. But a normal mouse will explore both, and then a mutant mouse, HOXB8 mutant, which is anxious, will only go into the dark one. Okay, here's the quantitation of that. So here we're looking at light dark, and how much time are you spending in the light? Normal, wild type, goes quite a bit of time, 60 seconds in a period of time. And then the other one that's a mutant spends much less time in the lighted box. So these are different ways of assaying anxiety. And by this measure, HOXB8 mutant mice are anxious. And they're chronically anxious. They're anxious all the time. Now, we also developed our own way of assessing. Okay, the other said that. We wanted to, one thing you would think about anxiety is if you were put in that situation and nothing happened, then you said, oh, okay, this doesn't seem so bad. You repeat it and nothing happens. It's even less anxious. And you repeat it 20 times, by that time it says, I'm not anxious at all. Okay? That's called habituation. Okay? And essentially it's an exponential curve, and you get more and more comfortable with the situation if nothing bad can be associated with it. It turns out that those other assays don't have habituation, and the reason is because they're complicated. But we wanted to develop an assay that was really simple, and then they would habituate. And so we did that, and what we do is simply fix the head, and that's, that's not happy with the mouse, and they're not used to having the head fixed, but there were reasons for doing that. So we have to habituate it for 3 days so they aren't anxious about that. And then what we do is ring a bell. And it's a sort of a loud bell, the sort of startle that makes it anxious. But if we do it repeatedly, pretty soon it doesn't care about the bell at all. Okay? And that's habituation. And that's shown, whoops. Okay, I wanted to give you one other aspect of it. I told you that the females removed their body hair much more aggressively than the males. Turns out the same thing holds for the anxiety. That is, if it's a mutant mouse, females are more anxious than males. And that's shown here in that open field. If we look at the females, they're staying in the closed compartment, and they're going very infrequently into the open compartment. Whereas if we look at males, and this is again a mutant, then they're still exploring both. So again, a sex association with anxiety as well as removal of body hair. Okay? And that's true in humans. Okay, and that's what I was going into next. That is, we develop an assay where we have the head fixed, ring a bell. Oh, thank you. Ring a bell and then have it react. And that's what's shown here. And the reason we fix the head is what's coming next. Well, I'm not going to show you what's coming next, but what we want to be able to do is to look at particular parts of the brain to see what part of the brain is doing their activity. And for that, we want the head to be fixed so we can steer and tactically put a needle right to a particular part of the brain and then assess what's happening. So that's why it's fixed. And this shows you that a HOXB8 mutant mouse is more anxious with respect to that particular assay. So the red is the mutant, and the wild type is the normal, is the black. And you can see it starts up higher, and then starts, as you repeat the exposure time after time, it gets less and less anxious with that. So that assay allows us then to study habituation as well as anxiety and association with sex. So, next we wanted to assess the new assay to make sure it really works. I mean, if we have a measure of anxiety and it has habituation, we want to make sure it works. So we can actually induce anxiety, and there requires our head to be fixed, the mouse's head to be fixed, not ours. There's a part of your brain that's, you know, it's what gets us always into trouble. It's the amygdala. Okay? It's always fighting with the frontal cortex, which is rational and telling you what you really should be doing, and the amygdala says, no, don't do that, do something else. And particularly pay attention if something's bad going to happen. And it's always worried. Okay? So we put this needle right into a particular part of the amygdala, put a little compound, and it's called urocortin, and there goes the needle into the brain, and then we can measure, that induces anxiety. Just chemically, all of a sudden this mouse is really induced because urocortin induces anxiety. And we see in males, we can do it to males, we can do it to females, and they react pretty much the same. We can also look at cortisol, which is the readout of anxiety, in the blood itself as a consequence of being anxious. You produce that chemical, and then it triggers off all the responses to anxiety throughout your body. Okay, so that's cortisol. We can measure that. And we see cortisol goes up, so that means that assay is working. And then we can do the actual experiment, ring the bell, and we can see in the presence of urocortin, and in the absence of it, and it really responds. The females really respond, the males really respond. So that says that our assay is working, and that we now have an assay for anxiety as well as habituation of the anxiety. And here we're looking at females, and they react. And here we're looking at, these are now mutant females, they react. We look at how much time they respond to cortisol. They're anxious because even though we're not injecting it, this is now the mouse is producing it because it's anxious, has high levels, but the males have higher levels, but not as much. And so again, this system is responding, this new system is responding to both being a female or a male, as well as anxiety, level of anxiety. Now, if we look at the time frame of when the mouse starts to develop this behavior, what we see is it takes off in about 6 weeks. And 6 weeks in a mouse is when it starts to sexually mature. This is puberty. Okay, 13 years in humans, 6 weeks in mice. That's why we work with mice. Much more. So here's the female, there's a male, and there's a normal. It's way down here, bottom of the curve. But this told us, well, maybe if it's responding just to sexual maturity, maybe it has something to do with sex hormones. Okay? So we wanted to test that. So is excessive grooming as a consequence of producing female hormones? And it is. We tested this 2 ways. We can take ovaries out. Okay, now you have no longer the capability of making sex hormones. Or we can put a chemical in, trilostane, which does the same thing. It inhibits the main enzymes that makes all the steroid hormones. Okay? And again, what we can see is in the presence of DMSO, this controls this a chemical, and then in the presence of trilostane, or if we have ovary sex demise, now if we look at how much coat load, it goes way down. If we look at coat loss, it goes way down. Here's again our heat map. DSMO, this is the mutant female, and high levels. Now trilostane treatment, this is over weeks, you give daily injections. Now the pattern goes back down to males. These are all females. But the pattern, the heat map, is just like a female. And if we ovaryrectomy, takes the ovaries away, then the pattern is back down to males. Okay? So that tells us that sex hormones are important in this process. And here we're looking at production of hormones. And we look at progesterone, which is a female hormone. Again, in the presence of ovarectomy or the males, that level goes way down. And similarly, if we look at another steroid, it goes way down. So it's reacting essentially as it should. And here we're comparing the behaviors. Females, DMSO, trilectin, ovarectomy, and males. And we can see now the behaviors are being rectified. Instead of having high levels of removing, staying in the enclosed platform. If we look at cortisol, reaction of anxiety, or if we look at the pupil of the not dilating, then again it's high. But if we treat either taking away the ovaries or with trilostane, then the levels go back down. Okay? So that says everything's under that control of the sex hormones. So it's harmony. So far, what I've showed you is, in summary, what we have is that Hoxby mutant mice. Okay? The only difference in that mouse is that one protein has been changed. It has compulsive hair removal, high levels of anxiety. Both are sex-linked in the females. And finally, the higher levels of pathology can be reduced by removing sex hormones in the female. So next, what we want to do is to see what is Hoxby doing and in what cells of the body, what cells are responsible. We think it has to do with the brain simply because all of these behaviors are controlled by our brain. Okay? So what we have to do is label our cells. Okay? Somehow we have to be able to see Hoxby cells. And what we do is label them either green or red. In this case, we're going to label them green. We use a gene that comes from a jellyfish that makes a product that fluoresces green wherever that product is being made. And so we put that under control of Hoxby. So wherever Hoxby is controlled, those cells will turn green, and then we can see them. Okay? Well, here's our trick. We take advantage of an enzyme called CRE. And what it does is to do a recombination. What it does is, there's a thing called LOXB and a LOXP, and they're lined up together, okay? And this enzyme brings those two together and then clips the DNA, and everything in between gets clipped out. So that's a way that we can inactivate a gene by controlling where CRE is made, okay? And we're going to make CRE only in genes that Hoxby is functional in. So it's only those cells that are going to turn green or red or whatever color we'd like, okay? So that's the technique. And so here is the way that we do it. We have one mouse that has Hoxby, and then it controls where CRE is made. Okay, so that mouse says, make only CRE in Hoxby expressing cells. And then we have another mouse which has a reporter gene, GFP, which is green fluorescence for a jellyfish. Okay, and then, but we put a stop sign in front of that gene. But then we flank that with those LOXP sites. CRE made over here will then take out that stop sign, and wherever CRE is made, then those cells will turn green because the stop sign is removed. Okay? That allows us to control gene activity in any cell type we want when we want it. Okay? And here we got an enormous surprise. We were expecting specific neurons in the brain to turn green. And what did we see instead? We saw things that look like microglia, and I'll come back to what microglia are. Those are those green cells. They're labeled. Okay? And one measure of what those cells are is they express a gene called CD11B, and those genes are only expressed in microglia, and they turn cells red. And here we're looking at coincidence, and you see all the green cells turn yellow. But it's not all the red cells that are turning yellow. Okay? It's a subpopulation. 30% of the cells are Hoxby8, have expressed Hoxby8, okay? Because that's where CRE was made, and they made those cells green, okay? So we know they're labeled as HOXP8, so they're the cells we're looking for in the brain. We completely, it was unexpected because we were expecting neurons, and we see microglia. And I'll come back to what microglia are. And this was a huge surprise, because microglia turn out to be immune cells. They're made in your hematopoietic cells. They're the same cells that are, where you make red blood cells that make your blood, so it can get hemoglobin and then oxygen. And they also make all the cells that are involved in mounting an immune response against pathogens. Okay? And microglia are, they're made by the hematopoietic system outside the brain. Okay? And they start out, I'll go through how you make those blood cells in just a minute. But here what we're seeing is microglia are controlling, these are immune cells in your brain, are controlling specific neural circuits that have a very specific output behavior. That is removal of body hair. Okay? Nobody would have believed that. Okay? And so we were really surprised. So that's what I'm saying. We're suggesting that microglia are controlling specific neural circuits that are, when they're not working right, give rise to a very specific behavior. Okay, but this made an enormous prediction. We can do bone marrow transplants. Okay, we can take radiation, destroy your immune system, and then put a new immune system back in it, and then you're functional. Okay, they do this for curing cancer, blood cancers. Okay, a person has blood cancers, they kill the immune system that the patient has, then it's supplemented with bone marrow that is compatible, hopefully a twin, that, if you're lucky, okay, and they happen to have a twin, and then they put them into you, and then you're cured, because now you have good bone marrow that's no longer cancerous. And so we're saying we can cure a neuropsychiatric disease with a bone marrow transplant. My students said, I'm going to do what? I said, you're going to do a bone marrow transplant on a mouse. And they said, okay. So this is the experiment, and here is the actual outcome. Now here what we're starting, first of all, we want to make sure that we could get cells in the brain. So we just took bone marrow that had been fluorescently labeled green, put it into the, irradiate the mouse, put these cells into the tail vein, actually, so they have to go through the whole blood system, finally get into the brain. Then we look in the brain, do we have green cells? And the answer is that, okay. And we actually, because we're lethally radiating the mouse, we get good uptake. If we didn't, the mouse would die because it wouldn't have any red blood cells. Okay? So the experiment has to work for the mouse to survive. We look in the brain, indeed we have green cells. That says that the experiment works in the sense that we can do a bone marrow transplant in a mouse. Okay? And here's the actual experiment. Here's a mutant mouse. It's already removed all its body hair. If I showed you the same mouse a week later, it would have lacerations all over its stomach. We do a bone marrow transplant, we radiate it, lethally radiate it, do a bone marrow transplant, look at it 3 months later, all the lesions are healed, the hair is grown back, and this mouse is permanently cured of that behavior. It no longer has the removal of the body hair, okay? By putting in normal bone marrow into that mouse. So, and that's shown over here, that's the mouse that has its hair grown back. And if we look at how much time they spent grooming, it's now back to normal. There's a wild-type mouse, a mutant mouse, and then if we've rescued it with the bone marrow transplant, then the behavior goes back to normal. Okay? And this mouse is permanently cured of that behavioral anomaly. Okay? We can obviously do the opposite experiment. We can take bad bone marrow, radiate a normal mouse, and give it the pathology with mutant bone marrow. Okay? So it's both necessary and sufficient to be causative. Okay. So that says everything's according to prediction. The bone marrow may be responsible for that pathology, because if we cure it with normal bone marrow, it's okay. If we give it bad bone marrow, we can cause the disease. So we make it, an inference that the bone marrow cells were causative for that disease. So now, where do microglia come from? And we had to address this because the literature says there's only a single source of microglia. It's in the yolk sac. Remember I told you hemopoiesis is very complicated. It starts in the yolk sac, then goes to the AGM, and then goes to fetal liver, and then goes finally to your bone marrow, and then it functions. And then some of the cells in the fetal liver, in the yolk sac, actually directly go into the brain, and those are the microglia. Okay, so in order to do these experiments, so this is what's in the literature, and they're very nice experiments, but I can tell you what's wrong with them, but it looks good, and that is that there's a single source of microglia, and they all come from the bone marrow. And that was published back in 2010. And it's a very pretty experiment, and that's shown here. So there's the placenta, there's the embryo developing, and the yolk sac around it, there's blood islands, and they directly go into the brain, and then cross the blood-brain barrier and go into the brain proper and become microglia. That's the, and what we're suggesting is there may be an independent source of microglia. So we wanted some way of following OXB8 progenitors, and we did this by making two-colored mice. So we labeled all microglia green, using a marker for all microglia, and then we made OXB8 microglia red, and then we could follow it, okay? So, and we did this using, again, this Krelok system that I just described, but now we're using two colors. So all microglia will be green, and then OXB8 microglia derived from a different cell population will be red, and they will become yellow. And again, what you can see down here is actually we're using two-photon microscopes in a live animal. So you're looking at microglia in a live animal, and what you can see, some of them are red, some of them are green, and then the red ones are also green, therefore they become yellow. Okay? And that's the population we're interested to see. Where do they come from? Now, there's lots of possibilities. One of them, come back. Okay. So what we did was simply look at red ones and green ones as a function of time. And if we look at about 9.5 gestation period of a mouse, instead of being 9 months, is 3 weeks. Okay. Again, while we work with mice, very quick. Sexual maturity is 4 weeks, okay? Really fast, woo. And then, so now we're looking in the brain. You see green ones, they start coming in at 9.5 days of gestation. The whole gestation period is 19 days, so about halfway through, we start seeing green cells in the brain. And then at 12.5 of gestation, now we start first seeing yellow ones. And then we see more and more yellow ones until we reach a plateau, which then is steady state, where about 30% are yellow, and then the rest are all green. Okay? So two types of microglia, and this could be accounted for in two ways. One is the green ones turn on the yellow gene, okay? So now they become yellow, okay? The other possibility is a different source of cells, and they come in later from somewhere else, besides the yolk sac. Okay? And how do we distinguish that? And I haven't told you something yet, and that is the color that we're looking at doesn't tell us when the gene is functioning. It simply tells that the gene functions. OXB8 had to be expressed in order to turn on the red fluorescence. Okay? But once it's on, those cells will be permanently red forever, and the gene could turn off, and the cells will still be red. Okay? And that's called a cell lineage marker. It labels those cells permanently, and we can follow them. Okay? So here we're looking, this could be that OXB8 is turning on late, or it could be red cells that get into the brain later. Both possibilities. But the way to distinguish that is to actually look at when the gene is actually functional. Okay? And for that we use what's called RT-PCR, where you're looking at the presence of RNA, what makes the protein at different functions of time. And what we can see in the yolk sac, there's lots of expression of OXB8 gene, and then it goes down very quickly. AGM, it goes up a little later, goes up, and then comes down. Fetal liver is almost not detectable. In the brain, we never see it expressed at all, ever. Okay? So that's important because that rules out the possibility that green cells in the brain turn yellow. Okay? That never happens. So it has to be another population of cells that are coming into the brain later time, okay? So, and then we can look at this by simply, here it's simply telling you again that hematopoiesis is complex, it goes from one stage to the next, and then finally we can look at these cells by simply sorting them, and looking for red cells and using a marker for stem cells, c-KIT, and what we can see is very few red cells are in the yolk sac, 3%. And then in the AGM, we see now lots more. And then in the fetal liver, a little bit of the delay, and then we see much more. It's about 16-fold more in the AGM than the fetal liver, and 180-fold greater than in the fetal liver than compared to the yolk sac. Okay? So not only are they going from one compartment to the next, but those populations are expanding, and expanding enormously, one step after the other. Okay? Here we're simply looking at percent, and again, what we see, very few yellow cells, progenitor cells to Hoxb cells in the yolk sac, lots more in the AGM, and going almost to 100%, and then finally in the fetal liver, going to 100% fairly quickly. And so this led us to the second model, where there has to be it's not just all microglia get into the brain at the yolk sac at E9.5, but there's a second population that appears to be born in the yolk sac, but then goes to the AGM, then the fetal liver gets expanded, and then finally goes into the brain starting at 12.5 gestation. So now we see this more complicated, things are happening, there are two populations, and they have different properties, because one is going directly to the brain, the other goes to a whole bunch of other places, getting signals to say be different. Okay, here you're a different population, and you're becoming different as you go throughout the body. So now we have two populations, and the question is that how similar or dissimilar are they? And are they really? Scientists are very skeptical. Particularly if you're telling them something that's completely wacky. Okay? Microglia are controlling neural circuits, and they have different sources instead of a single source, and so on. You're confronting the dogma. And so you have to be very careful to show, are these really microglia? Do they behave microglia? Do they look like microglia? And do they smell like microglia? And perhaps then they are microglia. So the first thing one looks at is simply genes. How many genes are different in one population? We can sort them. Ones are yellows and ones are green, only green. So we can sort them, separate them, and look at what genes they function. And they're actually, the main point of this is down here, you have 30,000 genes. So each little dot is a gene. Okay? And then we've expanded the size of that if it's different. And these are five-fold differences, from this here. Compared to these are all the same. Okay? So there are only 21 genes that are different, sufficiently different to be more than twofold difference in expression of a particular gene. So, one says, well, there's 21 genes different, but the other says out of 30,000, that's a very small number. So they are very much look like microglia in terms of gene expression pattern. Okay? That's the main role. But these genes are important. Those 21 genes make them different. Okay? And we know what those genes are, and we're publishing that, and so we can start asking what those genes are doing. Okay, so that's what SIMC says here. 21 genes are different, and they have interesting properties. Okay. Now there are two markers that everybody accepts as being markers for microglia. One's called TMEN 119, and the other CEL1. And since we've just looked now at those genes being expressed, we can, and in that profile, are those two genes expressed in those two populations? They're expressed at the same amount and so on? And the answer is yes. So by that criteria, which everybody says is the criteria for microglia, those genes are, those populations are also microglia. The Hoxb are microglia. Okay, so that's what this says. These are bona fide parenchymal microglia period. Now, because the one is red and one is green, we can also count them and see how many of the red ones also have team 119 and really are microglia, and the answer is greater than 98%. So all the red ones that we look at are indeed microglia. And now that allowed us to do a very important experiment. And that is to isolate those progenitor cells from fetal liver, okay, not the yolk sac, but the fetal liver, and then put them in the brain of a naive mouse. We've colored them so we can see them, and ask if they become microglia. Okay? Do they express T119? So we take fetal liver, isolate, and purify those cells out, transfer them, and put them directly into the brain of those progenitor cells, and ask if they become microglia, and the answer is yes. And here, those are the red cells. We sorted them out as red, but now they also express green, even though we didn't sort for green. And they also express T119. Not all the cells are T119 positive, because there are microglia still present when we inject it into the mouse that are not colored. Okay? So you expect that. And then the coincidence expression says that the green ones and the red ones are the same cells. And this allows us to do a lot of things now. Because remember, we've isolated those cells, we've purified those cells, we've put them back into the brain, and they function. Okay? So in the future, that'll allow us essentially a conduit to do anything we want to that poor brain in mice. Not very. Okay. So we've shown you that the progenitors are microglia. They behave like microglia. What do microglia do? They do a lot of things. And here's one experiment. We take a laser and we knock out, you can't see the neurons because we haven't labeled it, but you can see microglia, you can see red ones, green ones, and yellow ones. Okay? The green ones are all non-microglia, the yellow ones are hot-speed microglia. We're going to use a laser to kill a particular neuron right in the middle of the field. And then we want to watch the microglia and see how they react. Microglia know when a cell is in trouble, and they send the projections to that problem, and then they start doing something about it. Okay? What they do is to actually remove it if it's dead. Okay? So we're going to see a laser zap cells right in the middle. When I turn on the form, if it works, bam, there it is. There's the neuron dying, and all the cells are sending the projections to the site of injury. Yellow ones and green ones get there exactly at the same time. Bam! Now here go the projections right to the dying neuron, and they get there exactly at the same time. So by this criteria, they're behaving like microglia. We can do another thing. Remember, I can't stick a needle into the brain because then all the microglia go to the needle, okay? Because we're doing damage as we stick a needle in. So we have to do damage from the outside. So what we can do is cut the facial nerve. These are the nerves that allow you to kiss. They pucker out, okay? And you can cut them on just one side, but they're neurons, but the cell bodies are back in the brain. And if you cut the connection, then the cell bodies die, because now they know that they're no longer connected to muscles that allow you to pucker. Okay? So what we've done here, you're bilateral. So we've destroyed a nerve, facial nerve, on one side of the brain, and not the other. Okay? On that side, we didn't destroy it. On this side, we did. And we're looking at the region where these cell bodies are. And what you can see is microglia, the green ones and red ones, are all going right to that site of injury. On the other side, nothing's happening. Okay? And you can't distinguish red ones and green ones, but if you blow it up, you can see it, and you can count them. And indeed, the yellow ones and green ones are getting there at about the same time. But actually, the yellow ones are actually better at it. There are more of them, and they get there faster, okay, than the green ones are. So they're starting to distinguish in behavior. Now, now you can hear me. Another thing that microglia do, you make twice as many connections, neural connections, as you need. And then what happens is some of them are working and some don't. And then there's a mysterious process that says, get rid of the ones that have weak connections and keep the strong ones. And this is what's called pruning. It's about 50%, right? And it's a rule, no matter what kind of connections you're talking about, you make twice as many, the ones that are active don't get killed, and the ones that are inactive get killed, and then thereby strengthen the ones that are active. Okay? And microglia turn out to be important for that process. They can recognize which are the strong connections, activity dependent, and then they go there and remove the ones that are weak and leave the strong ones. And that's called synaptic pruning. Okay? And one way we can follow that is to stick a dye in the eye, and then it goes back along the processes, back to the connections in the brain, and then that dye sticks to synapses. Okay? And it's a yellow dye, and so you can see them, and then you can ask, do microglia phagocytize those weak connections? Are those pieces of synapses inside the microglia as they're eating them? Okay? So we look at that, and here we're going into the brain. We've injected the dye. We go to the thalamus and look in the thalamus and see whether we can see microglia and whether they have things inside them. And there you have two microglia, a red one and a green one, and you can see the little dots. That's the pieces of synapse that are sticky, that used to be part of the connections, and it's phagocytizing. But if we quantitate the yellow ones and green ones, and they're equally active in the ability to synapse. And we can do contralateral, we can inject dye just on one side, and look at the opposite side, and then the other side is the control. Okay? So that tells us these microglia are just as good, the HOXB8 ones are just as good as the non-HOXB8 one in terms of synaptic pruning. Okay. Now, so far I've showed you everything's the same, but they have to be different. And how are they different? And that is interesting. Here we're looking at the size of the brain. Okay? Anterior is at that end, posterior, the cerebellum is at this end, and red label is the microglia. And then also, if you look individually, you'll see red and green microglia all throughout the brain, and then you can start counting. And where are they? And they turn out that HOXB8 are in one region, and all the non-HOXB8 are in a different region. So they know to be in different regions of the brain. Okay? How they know that? I'm not sure, but we can find out. Okay? What is telling them to go in the right place? And why is that the right place? Here it is, the motor cortex involved in movement, the anterior aspect of the striatum, and the lower of the lateral orbital cortex. That region, which is high in HOXB8 microglia, is what's called the Ocd circuit. These are MRI scans in humans, telling you the patients with OCD and trichotillomania, that region of the brain is overactive. And it's been defined as the Ocd circuit, and it's where the HOXB8 microglia are. Okay? So they're in the right place to activate or modulate the right circuits. Okay? So that's an interesting observation. All the other red is because there are projections coming from the spinal cord. It has nothing to do with our other story in the cerebellum and so on. Okay? But HOXB8 microglia are in the right place to do what they should be doing. Now we can do one more thing about finding out whether those genes, those cells, are really responsible for the behavior. And what we can do is what we call a cell ablation. We can remove all HOXB8 microglia from the brain and then ask what happens to that mouse. Okay? And how do we do that? That's shown here. It's again using Prelox. Okay? And we have a gene called CSF1R. Colony stimulating factor 1 receptor. Okay? Turns out that that gene is important for the survival of all microglia. So if we remove that gene, then those cells die. Okay? So what we're going to do is use Prelox to inactivate this gene only in HOXB8 cells. Okay? That's HOXB8 pre. Only HOXB8 cells will have pre. And then we put, instead of having a color, now we put this gene under the control of what pre does. Removes the gene in the presence of pre, because it has LOXP sites on either side of that gene. Okay, so we put LOXP sites on either side of CSF-1, made with a mouse that makes FOXP83. Only those cells will be dead because they don't have that gene. Then we can ask the consequences of what happens when you don't have those cells present. Okay? And then here we should look at the experiments. We see red cells, those are the FOXP8 microglia. We look after we activate CRE, we activate CRE in those cells. All the microglia are on, red ones. The green ones aren't affected, they're there. And I'll show you that in the next slide. And we can quantitate it and show that greater than 90% of the cells of FOXP8 microglia are dead. Here we're looking at all cells, and in terms, so up above is without the gene being activated, down below is with the gene activated, and you can see a lot more cells. And in fact, what happens is this body already knows that you've killed all the red ones, and therefore they say divide, but there are only green ones there to divide, and they fill up the space and compensate for killing the red ones. So that's why at the bottom you see actually even more green cells than before. Okay, because now the 30% that used to be FOXP8 microglia are no longer there and are filled with now non-FOXP8 microglia, which are green. Okay, so that says the experiment works, and here's the quantitation. We can do that in a normal mouse, or a FOXP8 mouse. And then next we actually do the experiment. What happened? Okay. So first what we did is eliminate it from FOXP8 microglia themselves. That is a mutant mouse. Take away FOXP8 microglia, ask what happens, and the answer is nothing happens. Females, males, we're looking at anxiety levels, we're looking at loss of code, here we're looking at cortisol levels, anxiety levels, and response to dilation in the presence of sound. Okay, and nothing happens. What happens when you knock out FOXP8 cells in a normal mouse? Completely normal mouse. Okay, that's shown in the next slide. Here. Okay, this is a non-FOXP8 microglia. We've just removed that one gene in FOXP8 cells, and now it develops a pathology. Okay, that says that the FOXP8 microglia, the cells themselves, not a mutant, normal cells, we take them away, and now the mouse has it. That says that those cells are preventing having that pathology. People always said that microglia caused the sensory anxiety, caused inflammation in the brain, and I'm saying they're doing good things. They're preventing all of those things from happening. And you can just remove the cells, and here now is female specific. If you look at, and if you inactivate those things, you see in the absence, you see the male pattern. And so it's not, it's sex dependent, and it's also only dependent on having those cells present. And that's shown here. Here's females. In the presence of removing that gene, you start removing code compared to a control. It doesn't remove anything. If you look at the amount of grooming, it goes up. And in males, they react, but not as much. Okay? And that's shown here. So here we're looking at the females reacting and not the males. So in summary, what I've showed you is that eliminating the HOX gene does have an effect, and both in that having the mutations has pathological grooming as well as anxiety-related behavior, that the eliminating microglia in a normal mice recapitulates that hypothesis, therefore must be responsible, and that microglia are doing good things normally, unless they're mutant, and we can conclude that the role of normal microglia is to prevent and protect against anxiety, and then if you have chronic anxiety, then you have pathological effects in terms of OCD and OCD spectrum disorders. Okay, and further, that non-FOXP8 microglia can provide this function, because if we have all the microglia be green, it doesn't help. You still get the pathology, so they can correct it. Then finally, it gives us, now we have all the tools to actually figure out what is really happening. That says microglia have to be talking to neurons. What is the language? Okay, and now we're in a perfect position to be able to address those kind of questions. There's a technology called optogenetics. It's actually taking a channel from an organism that actually activates those channels by light, the sun being on, and then that makes essentially that pump generate pump sodium. Alternatively, you can make it pump a different channel, pump chloride, and it inactivates the neurons. And the remarkable thing about it is because it's light dependent, you can turn it on and off the light, and it's milliseconds that those reactions take place. And if you have something that you can turn off and on in millisecond time, you can generate a pattern. Okay, you can make a Morse code. Okay, and that's information. And so then you can ask all of a sudden, if you do that in one cell type, does the other cell type respond in the same frequency? And if it does, they've got to be talking. And what is that language? And so once you understand that they're talking, then we can start to figure out the language. That is, how can microglia be possibly controlling neural circuits? And I think there are many possibilities. So here's our microglia. This is a circuit that's important. That's the Ocd circuit, Transplants circuit. And the question is, how do microglia talk to this circuit and modulate its activity? It could be cytokines, it could be chemical. There's something even more interesting. These microglia have these projections that are waving in space all the time. And they're really rapidly. And what you can figure out, because there's lots of microglia in the brain, they can actually cover the entire brain surface in a period of 30 minutes. Okay? So they can assess what's going on in the whole brain surface. But what you also see is all of a sudden they stop. And then you can ask, what are they doing when they're stopping? What they're doing is they're putting the projections right across a synapse. They completely surround that synapse. And that changes the property of synapses, changes the conductivity across that synapse, and therefore could modulate neural activity. It could be as simple as that. So, but we have now the tools to start asking that, and what I'm hoping that somebody out there finds that fascinating and comes and works in my lab. So I think we're in a new age. We have cells that we can start manipulating. If you have a neuropsychiatric disorder and somebody tells you, I'm going to change that circuit, you better laugh. It's really complicated. But now we have cells that are there. 90% of the cells in your brain are glia. They're not neurons. 10% of your brain is neurons. The rest is all glia. So it's that population that you really want to be manipulating and be able to do things that they weren't doing before. We can take them out, manipulate it, and put them back in. Okay? So now you can start your imagination of the kind of things that will be happening 10 to 20 years from now. Thank you for your patience.
Everybody can take a seat real quick. We have a few minutes for questions, probably about 10 minutes. We're running a little over on time because we started a little late. We have two chaperones that are going to be running around with microphones. If you'd like to flag them down with your hand, we can ask questions in pairs like that. Does anybody have any questions at this time? Any questions? Do I see any? I see one over there.
Hey, sir. I'm curious. You know how you were talking about how you took out the bone marrow out of the mouse and did radiation? Do you think you can use that for say other diseases or viruses and cure them by transplanting another person's bone marrow into them for cure?
No, I think initially people were afraid to do that. Yeah. So the question is bone marrow being used for other types of diseases? And it is being used, for example, with cancer. If they have a blood cancer, you can irradiate and then put bone marrow back that isn't cancerous, and that works. More interestingly, it's been done with AIDS. Okay, So what happens is that 1% of people are actually resistant to AIDS. So you can irradiate the person with AIDS and then put in compatible bone marrow that doesn't have AIDS and is resistant to the virus, and that cures that patient. Okay. Now, I'll tell you one more story, and that is that there are about 600,000 bone marrow transplants have been done. Now, I've told you that these diseases occur at about 3%. So a lot of those patients had OCD and had trichotillomania, okay? Just randomly. So I've called up bone marrow centers, first of all, and asked, have you seen anything funny after a bone marrow transplant? And they said, well, we have been looking. I said, you better start looking. Then I go to the literature, and indeed people have done this experiment in both directions. They've taken bone marrow from a patient that had OCD or trichotillomania, put it into their brother that didn't have it, and gave it to them. Okay. Vice versa. They've taken bone marrow, a patient that had trichotillomania, and put in bone marrow that didn't, from a normal patient, and they were cured. Because that experiment has already been done. Okay. So that says the bone marrow transplant could be used for neuropsychiatric disorders, but right now about 2% of people die of a bone marrow transplant. However, people are working on techniques to make it so that a lot fewer patients die. And then once you have those conditions, then I think you will start seeing that. Okay? And this would be true for a lot of diseases, not just what I've showed you. Yeah.
That was magnificent.
No, thank you.
My question is, I'm familiar with the research by Dr. Marrow, which is the nature of consciousness and the structure of the microtubules and the neurons. My question is are you looking at the internal atomic structure of the glial systems?
Yes.
And if they have a microtubule or kind of a graphene type of structure with carbon?
Right. Okay. No. First of all, the microglia do have microtubules, but they're not neuronal type microtubules, which are different from other cell types. They're like other cell types because they're hematopoietic stem cells. So in that sense, they are different from neurons. Right now, what we're really trying to address is further out. That is, what are the genes doing? What can we do about the pathology? And then later, we'll get more molecular and then submolecular, as you suggested. So I think that's way in the future. Right now, we're still sticking to essentially, trying to understand how the system works and how can we manipulate it in terms of therapy. A question over here.
Hello. My question's on the microbiology. I'm not familiar with biology. I'm a biology major myself, but thinking upon it, I've done some research into Alzheimer's. It runs my family a little bit. And I was curious if your thoughts on microbiology being directly associated with them not being able to remove dead cells or the such, and then HOXB8 could be similar to their function of removing dead cells or helping with the brain stabilize.
I'm sorry, I didn't quite catch the question.
Do microbiology help with Alzheimer's and HOXB8?
Does it affect other diseases? Yeah. No, first of all, I should say there's enormous overlap between the patients, for example, that can't communicate in terms of with each other. Okay. So, and our feeling is that that chronic anxiety, that once you have chronic anxiety, that can lead to a lot of different neuropsychiatric disorders outputs. And the outputs will, in the case of the mouse, was trichotillomania. And we also have connections now with humans in the sense that, one of the questions you could have asked is, well, you've shown us everything in mouse. What about humans? Okay, do they have differences in OXB8 gene to begin with? And does that difference lead to the same pathology? And the answer is yes. They have, obviously they have OXB8, because I told you all the genes are almost the same. But furthermore, they have changes in that gene that also track with the disease. Okay, so that says that there's a connection in humans with respect to the OXB8 mutation as well as the pathology. So that closes the circle, that it's the same system. But what I'm saying is that I think it's much broader. I don't think, if you ask me, this whole system was set up just to control OCD or trichotillomania. It doesn't make sense to me that you have such a bizarre, big system to control one disease. I think it's much broader than that, and our outlook is that what's at the root is actually anxiety, and these mice are anxious. And then chronic anxiety had different outputs depending on your environment, that is your history, your total history, as well as your genetics. So then the output will be different depending on how those two factors enter in. In some cases it will be trichotillomania, in other cases it may be OCD or Alzheimer's disease or whatever. Okay, so that it's the chronic anxiety that's actually at the root of all this, and that's what the system is involved in regulating that anxiety and trying to keep that anxiety under control, and that then leads to multiple neuropsychiatric disorders. Yes?
Okay, mine's pretty simple. It's two questions. One, how long did it take to kind of make this conclusion? How many years of study? And then, where does it go from here? Like, what is the next step with this information?
Sure. It took a long time, but not for the reason you might think. I have a policy in my lab that if a student works on something, or a postdoctoral student who works on something, and they then like the project, they can take it with them. Okay, and the reason I'm doing that is because then I know I don't have to worry about it. It's in good hands, they'll do it, and then I'll do something else, okay? And so the student that was working, that initially made these mutants, wanted it. I said, so it's yours, okay? But then she decided she really didn't want to work on that later. I mean, she was a student, then had a postdoc, and by that time she decided she wanted to work on something else. So then, 8 years later, I asked her, are you still working on this? She said, no, we never got started. And I said, well, we'll take it over. So we started again. So there's an 8-year lag, nothing was happening. And then all of a sudden we started getting interested again. And then we discovered there was microglia, and that's very quick. And that's just a few years now. And I think it's still steep rolling ahead, I hope.
I think we have time for one more question.
Doctor, is there a difference between anxiety that is perhaps genetic and inherited or something that is caused by trauma?
Yeah. I think the source of the anxiety can be different. And it can be a specific trauma, it can be many things that can cause the... And it is important to know what the source of that leads to anxiety. So it could be external, but it can also be internal. And in this case, a OXB8 mutation gives rise to high levels of anxiety. I mean, that mouse, it's none fault of his. We made the mutation, and breed them, and they all have it. So you can have both internal effects as well as external. And the question is, are they same? And I think they're activating the same axis. They essentially, the hypothalamus, pituitary, and adrenal axis. So that axis is being activated in both cases, and then that makes cortisol, and then high levels of that leads to anxiety. So I think the end root is the same, but it can be from different sources. And it's again, nature is amazing. I mean, there's a system, you all know about it, the hippocampus. And it's involved in telling you where things are in the world. It orients you, it tells you, you start seeing familiarity, patterns in the environment you're in, and then from then on you remember that, and that you see it. Now, right associated with the hippocampus is another layer of cells, and they have what are called anxiety cells in there. Okay? So why would you want to do that? And that is because nature thought it would be clever if you associated not only the marks of an environment, but whether you liked it or didn't like it. Okay? So you want some way of the amygdala to get involved, saying, put a value on what you're looking at and assessing, is this a friendly place or is this not a friendly place? But it does that by juxtaposing those cells right next to each other. One layer of separation. Okay? So, it's a sense, it's because it worked. And because those organisms survived that happened. But it's a remarkable, again, story in the sense of, not only do we see things, but we also then color them with memories that are good or bad. And if we like them, we go back there. If we don't like them, we stay away from there.
Could I have a round of applause for Dr.